So for those of you that don't know me, my name's Kelly Lambert, and I'm an Associate Professor here in the discipline of Nutrition and Dietetics in the School of Medical, Indigenous and Health Sciences at the University of Wollongong. And it's my pleasure to welcome each and every one of you here today. For those who haven't joined before, the Luminaries series brings together leading researchers, industry experts and thought leaders for a 1-hour conversation every fortnight. And we're going to talk about research and collaboration and how the University of Wollongong is tackling global challenges. Today, I'd like to welcome colleagues and collaborators as we discuss how our current research is improving our understanding between diet, the gut and inflammation and the microbiome. Before we start though, I'd like to acknowledge country, and officially on behalf of the university, I would like to acknowledge that country for Aboriginal peoples is an interconnected set of ancient and sophisticated relationships. The University of Wollongong spreads across many interrelated Aboriginal countries that are bound by this sacred landscape and intimate relationship with that landscape since creation. From Sydney to the Southern Highlands to the South Coast, from freshwater to bitter water to salt, from city to urban and to rural. The University of Wollongong acknowledges the devastating impact of colonisation on our campus's footprint and commit ourselves to truth-telling, healing and education. Thank you very much for those who have joined us. With that introduction done, I would like to welcome our panellists today. So you can see 3 faces on the screen here. We have Professor Georgina Hold, and Georgina is a Professor of Gut Health at St George and Sutherland Clinical School, based at St George Hospital in Sydney. We have Chiara Miglioretto, who's an accredited practicing dietitian and a PhD candidate here at the University of Wollongong. And we've got Denelle Cosier, who's also an accredited practicing dietitian and PhD candidate at UAW. I'd like to encourage anyone that's online today to submit their own questions to us using the Q&A function, and I will ask our panellists those questions. We'll try to get through as many as we can, but if there are some remaining, I'm happy to provide answers after the webinar. We will receive a recording of this webinar at the end as well. So we might get started on the presentation today. So I'm going to hand it over to you, George. We're going to start with you. And while we're setting up that screen with your presentation slides, can you tell us a little bit about your work at the Microbiome Research Centre?
Thank you so much, Kelly. Good afternoon, everybody. It's an absolute pleasure to be able to join you today. I am a gut microbial expert. I've been practicing in the field of gut microbiology for since last century, actually, so makes me feel very, very old. But I was given the task of giving you some information about gut microbes, and just generally gut health and how we've come to be where we are in this relationship with our microbiome. So my talk is entitled Microbes, the Good, the Bad and the Ugly, and hopefully we can put some context to that. I don't know that I can share slides yet. Okay, here goes. I can now share slides. Okay, so I'm based at, I'm part of University of New South Wales. I moved over here from the UK about 5-6 years ago now. And I came over to join Australia's first microbiome research centre, which is based at St George Hospital in Cosier. So the Microbiome Research Centre focuses on all things microbiome and health related. It's a large translational world-class laboratory, which I had the pleasure of designing from the floor up, really. And it's there to really allow us to explore the world of microbes and the influence that they have on us. So the microbes that associate with us as humans, we define as the microbiome, and the definition of the microbiome is the ecological community of commensal, symbiotic and pathogenic microorganisms that literally share our body space. So that's really where the good, the bad and the ugly comes from. Although, you know, just as in life, good doesn't always mean it's going to be good and bad doesn't always mean it's going to be bad. There has to be context to that in terms of the external factors which influence the situation. So The one thing which kind of, always blows my mind about microbes and the influence they have on us is basically numerical dominance of microbial beings associated with us as humans. So if you look at it on a cell per cell basis, the anticipation is that we have more microbial cells than human cells. So we are probably about less than half of our 50% of us is human cells. The rest is microbial. But if you actually look at it on what each of the contributions of the different microbes versus the contribution of our genetic material, we're probably only about 1% human compared to the influence that microbial genetics can have over us. And when you think of it like that, it's kind of incredibly humbling, but also explains an awful lot about why it has such an influence. They have such an influence on our health. So we're really entering into the second decade, only the second decade, in terms of our understanding of the human microbiota, particularly the gut microbiota. And so this panel of publications that came up in 2012, and I bear no apology for using what we would now consider to be outdated, 11 years old, but this really set us on our journey of understanding microbes, understanding that they are niche specific habitats, that we can define and identify microbial or samples based on the microbes that are present in them. And so this is really showing you that based on body site, there are different organisms, microorganisms that are present there. And that makes sense because we know all of these different niches have a very different physiology. So not everybody would want to live in the same place at the same time. But where did it come from? Well, there's this concept that before you're born or at the point of birth that you are, that a baby is born sterile. Well, this is, I think we've demonstrated sufficiently now that this isn't actually true. But the things that happen in the very first few days, weeks, months of life have a really strong influence on the types of microbes that become associated with us. So the mode of delivery, whether it's a vaginal versus a natural delivery, the way that the child is fed, whether it be breast milk or formula fed, the interactions with family members, with pets, with the time that children are weaned onto solid food, all of these shape your microbiome and kind of set you up for life. So your microbiome pretty much stabilizes post-weaning. There are aging impacts on microbiome that go hand in hand with changes in our immune status as that develops as well. And we're also now starting to understand that health status, the impact on health because of your microbes is really, really important. And the fact that there are things that we can actually do to actually make us remain or to restore our health. And that's really what I want you to take away from today. So going back to that transformational paper back in 2022, this is showing you microbial diversity of different body sites. And all you need to take from this is that colors mean different groups of bacteria. So if you look at the different body sites, you can see that what's present in stool is a lot of blue and green with a little bit of yellow. If you look in the nose, the first panel, for example, the anterior nares, you see there's a lot of red and yellow versus green. It's just essentially showing you that difference between places. And that's on who is there. If you actually look functionally what they are capable of doing, you actually see a flat line over the different sites. So you can differentiate based on the species that are there, but not on the metabolic functions that the microbes can do. So what that tells us is that lots of different bugs are capable of doing similar functions. And that's really important because it demonstrates to you that you have to know what technique to look at your data to actually find the meaning that you need. So we know that metabolic and molecular functions are not species driven, but we also know that microbiomes change over time, again, because of based on their environment. And if we want to understand things like health, we have to look at things over time. And if we want to understand what's going on, we have to standardize, whether it be in collection protocols, extraction protocols, or analytical protocols. All of these things can impact on the data that we get out, and we need to have that engagement, that collaboration with our clinical colleagues so that we can make sure we have clearly focused clinical questions. So what is a healthy microbiome then? Well, again, niche specific. In some parts of the body, a healthy microbiome is a very varied and rich diversity of microbes. In other places, a healthy microbiome is a very minimal richness of a microbiome. What else is important? The environment, whether it be geography. We know that where you live, what you eat, what you're exposed to, and what your genetics are, all influence your microbiome. But probably what's most important is what's in panel D there, capacity and dynamics. How does that microbiome work? Does it work as a good functioning pack? Can they, members of the pack help each others? If you see, if they are subjected to an environmental insult, can they return to normal? These are the types of factors which we define in terms of understanding health and the health of a healthy microbiome. What do these microbes do for us? Why are they so important? Well, they digest the food that you eat. Good bacteria can split food into its nutrient components and release those factors to be available to us. We need microbes to be able to release vitamins from our food and synthesize things like vitamin K that we actually as humans can't do without microbes. And they also do things like detoxifying some of the nutritional chemicals that we consume. Good bacteria can also prevent the bad bacteria from colonizing the gut as well. So they do many, many functions for us. But they also do have an impact on health. And we know that microbes, certain microbes are associated with a variety of conditions, whether it be intestinal disorders, which makes sense because that's where the bugs are, such as inflammatory bowel disease, which I work on, whether it be liver disease, whether it be diabetes, metabolic syndrome, atherosclerosis, neurological disorders, cancers, but it also impacts on healthy states as well, such as pregnancy, and also things like joint disease. So all of these factors we now know have a microbial component to them. What does normal mean? Well, it depends on where you are. This is a publication from a few years ago looking at the microbial status of 3 to 4 month olds in various parts of the world, and just looking at those pie charts and seeing the colors differ indicate to you that the microbiome of healthy babies changes dramatically where you live in the world. So what this tells us is that if we want to look at anything to do with disease or health, we have to look in the right study for the geography that you're in. So there's no point looking at what goes on in Canada, for example, and comparing the healthy cohort from Canada to a disease cohort in Armenia and saying, well, it's different. Well, it's different because of geography, never mind the disease. So that's a really, really important point that we need to take home. You've got to understand what it is you're looking for. I just want to talk to you a little bit about inflammatory bowel disease. It affects a number of people in Australia. It's very much involved in affecting quality of life, and it has a huge impact on people. There are a number of overlapping influences in inflammatory bowel disease, and if you want to understand the disease, such as IBD, which is multifactorial, you need to look at lots of different things at once. So we set up the Australian IBD microbiome study to look at disease over time, to look at national and regional variations, to look at the microbial profiles of people who responded to therapeutics or didn't respond, because we want to look for designing new therapeutics. So the AIM study was launched in 2019. It's in multiple hospitals across Australia, and we aim to recruit 1,000 participants within the first 5 years to define the microbiota signature of IBD, and as I said, to look at things like medication and lifestyle factors. This is what the study actually looks like. we're recruiting, and this is the factors that we are actually collecting. So microbiome, host factors, patient-reported outcome measures, dietary factors, disease-related information, clinical history, and serial biological samples, as well as COVID history we added to this. And this all feeds into a multidisciplinary program of work looking at various factors, which I'm not going to cover now, and I'm happy to talk to anybody about if they're interested. But just to tell you what AIM has done since it was launched in 2019, we've recruited 950 participants. 593 of these are IBD patients, 367 of these are healthy controls or family members of IBD patients. We have 132 pediatric recruits into the study. Anybody can join the study as long as they meet some very soft inclusion-exclusion criteria, and they reside within Australia. And if anybody is interested in joining the study, then please scan the QR code. That will take you through to a registration page, and we'll be happy to answer any questions that you have. I'm going to finish my presentation there and hand back to Kelly.
Thanks, George. We've got a few minutes for questions, so I might ask you a question. What do you think is one of the most common misconceptions about the microbiome that you hear around the place that you'd like people to know?
I think my biggest, what I would really like to put out there is that looking at defining your microbiome is ready to influence any treatment that you have for whatever. So we are, as a collective, not in a position where understanding microbiome reports is going to be able to, in most cases, influence treatment. And our clinical colleagues are not well enough equipped to be able to look at a microbiome report that you may have, that you've got from a commercial company, and be able to alter your management based on that information.
Yeah, that's a good point. It's a waste of money in some ways. What do you think then, if you had to crystal ball gaze into the future, is that where precision medicine might be heading? You think that there's potential for that?
Absolutely. So, I mean, there's two things to say. One, I'm currently working on an international consensus to actually define what about a microbiome report should actually be contained within that report, what parameters, what definitions, and what should be the expertise of the people generating those reports and the information feeding out of that. I would hope that the publication about this will come out later on this year, but it's been a very interesting collaboration across the globe to get to the position where we are now, and we're almost ready to submit that for publication. The second thing I would say, and people maybe aren't aware of this, is that the microbes inside us also have the potential to influence our therapeutic outcomes. So, and we know this not just from inflammatory bowel disease, but from a number of other points of requiring therapeutics within health. We can actually profile your microbes and start to make decisions or judgments on whether you can respond to therapeutics or not. And we can actually, in experimental models, have been able to manipulate your microbiome before you take a therapeutic and actually switch you from a therapeutic non-responder to a responder. And I think that demonstrates the power of the microbiome. They're causing the problem, but also they are potentially the solution to our problem. And I think precision medicine is absolutely where we're going to go. And but we need to go there in a well-informed way, and we need to be ready. And at the moment, we're still learning to walk. We're not ready to run.
Yeah, it's amazing. Can I ask you one other question? If other people have questions, please put them in the Q&A. So it's the age-old question about probiotics.
Okay.
Should people take them? If so, when? If not, why?
Great question. I think my view on probiotics is that I think the concept is excellent. I think our knowledge in the last 5 years or so has shifted dramatically from what I will class as traditional probiotics, which actually didn't have as much information around them as we have now. Probiotics in the past were things that could be cultured and were considered not to do harm as much as they were to do good. What I think where we are now with your new generation probiotics is a much more carefully selected, much more sensitive microbes that have got clear demonstration on many, many levels of their ability to improve health. And so I don't dismiss existing probiotics, but I think that there's much more potential to come from the new age probiotics. They're just not quite ready for market yet in all cases, but they're coming.
Yeah, definitely. The evidence that we have from a dietetic perspective is there's strain-specific benefits, if there are any, and they're disease-specific. So just buying anyone off the shelf and taking it is of no use to...
Well, that's right. I mean, I would say one shoe does not fit all. And that's where we're going. And that's why it has to be down the precision medicine base. And I predict that in the future, just like you get for inflammatory bowel disease, you get your fecal calprotectin measured, for example. I predict that we will be submitting stool samples, and then your probiotic requirement will be defined, you know, in a way such as on the basis of a stool sample. And you will then walk out of the pharmacy with your own sort of cocktail that works for you but isn't going to work for somebody else.
Yeah, that's amazing. Okay, so thank you, George, for that presentation and the answers to those excellent questions we've all been wanting to ask. We might move now on to Chiara, if that's okay. So, Chiara, your presentation.
Lovely. Oh, I can see it. Beautiful. I'll have a little trial to see if this light work. If I can move. Let me have radio. I think they do. Yeah, perfect. Alrighty, good afternoon, everyone. It's very exciting to be here and to be talking about my journey into IBD. Obviously, it's not really as exciting as Georgina, which I love the topic, but the reason why I jumped on this is because I have a personal connection to it. And as Kelly started at the start, I'm also a dietitian. So I love food and I love sharing food with others. But I also know that for someone with IBD or inflammatory bowel disease is not always the case. Hence why the title of today's presentation is Opportunities to Improve the Dietary Knowledge of Australians with Inflammatory Bowel Disease. And so, sorry, it's not changing. Oh, there we go. So really, we know that information about what people want to know can be a good driver to engage the person in their care. But what we don't know about, what is quite limited, is their dietary information needs. So what is that people with IBD want to know when it comes to diet? We know the current evidence on managing IBD through nutrition focuses mostly on the acute end of the IBD journey, but it's not as much on just managing symptoms. And Denelle will touch on diet more shortly anyway. So there is guidance from your patient organizations like the Crohn's and Colitis Australia, which has a lovely website. However, there is also an abundance of anecdotal or misleading advice available on the internet, which can make things really confusing. And so what also emerged is that there is lack of guidance also from healthcare professionals like gastroenterologists or non-other dietetic health professionals. And so the idea is, sorry, it's not changing for some reason. I'm not sure why. Sorry, guys, I'll be 1 second. Okay, that's better. So basically, the lack of reputable and consistent information and the lack of guidance from healthcare providers when it comes to diet can lead, with no surprise, to people adopting a trial and error approach, which it's to improve symptoms, stay longer in remission, avoid flares, and things like that. And it's understandable that people want to do that because we know that there's lots of different diet therapies that promote healing your gut or improve your gut health. And today's talk is all about gut health. and lots of questions about prebiotics and so on. However, we know that this trial and error leading to food restriction can be a problem in terms of avoiding entire food groups. And as you see on the screen, there's this little graph that shows the foods that are mostly avoided by people either with Crohn's disease or ulcerative colitis. And attached to these foods that are also the individual nutrients that come with them. So as a dietitian, to me, this is concerning, because when we exclude a food group, or even just say dairy or red meat, we are also excluding nutrients that come with it. And so the idea is that, again, sorry guys, I'm having trouble with the slides today. The idea is that if we empower the person with IBD with enough knowledge, we are not only just going to support equal partnership with the healthcare provider, but we can also improve self-management and health outcomes, which in the case of IBD could be saying reducing the risk of nutritional complications, but also maintaining maintenance for a little bit longer. We know, though, as you can see from this puzzle, that IBD management is not just medication, it's not just diet, it's a puzzle. And we know that people with IBD want to know about diet, which is part of the puzzle. And so part of this PhD, as you can see here on the screen, there is the journey that I'm trying to complete. And the first study really was a review of what is the dietary information needs of people with IBD and where are people looking for this information. And so I'll jump straight into that study because it's been completed. Basically, what we found is that, yes, of course, dietary information is of interest. And we also found that people value it even more than medical therapy, with some people perceiving that diet can give them a sense of control in this otherwise relapsing remitting condition. The other thing is that the relevance of dietary information is shaped by the stage of the disease. And by that, I mean from pre-diagnosis through to diagnosis to remission and to flare. So at all these different time points, the information wanted can change. And that takes me to the timing of receiving information. We found through the review that diagnosis and flare were the time points where information was most wanted. And that the gastro, Dr. Google, Facebook groups, non-professionals like friends, family members, naturopaths, and to a minor extent, dietitians and general practitioners were the people tasked to answer certain questions. People also reported feeling that that piece of the puzzle, diet, was not well addressed throughout their disease journey. And so what are people actually wanting to know? So it's no surprise that the question of what should I eat or what should I avoid was quite prevalent. But there were also questions around how to maintain nutritional adequacy, taking supplements, and that could include, your prebiotics or your probiotics, and then generally healthy eating for IBD specifically. So then, because the questions that we found were generally broad, we thought, let's interview some people with IBD, but also healthcare professionals. And so far we interviewed about 32 people with IBD, 26 people with IBD and a handful of both dietitians working in IBD and gastroenterologists working in IBD. And these results are preliminary, so mind the lack of detail, but I'm still working on this. But basically what we understood is that diet is a very individual, but also very lonely journey, unfortunately. And that of those 5 or 6 questions that we found from the Scorpion review, there were another 350 that related to diet. And things that people were wanting to know are like, and Georgina touched on this, is there a probiotic that's better than another to improve my gut health? How long should I take it for before I see some results? Or if there is a food that is a prebiotic, how long should I take this for in order to see, again, some results? Then in terms of the timing that we briefly discussed on, yes, of course, diagnosis and flare again came across as really important time points, but also having checkups regularly, not just a one-off appointment that you might get after 3 years of being diagnosed. Pre-pregnancy or thinking of conception were also important time points, as together with the changes in medical therapy. What we also introduced in the interviews was a diet-specific tool for IBD, which we will be working on, so watch this space. And then last but not last, we are still recruiting some people with IBD, particularly males with either Crohn's disease or ulcerative colitis. So if you want to click on the QR code or take a picture, that would be really handy. And that's me for today. Thank you so much.
Thanks, Chiara. Can I just ask you a quick question about if there are people in the audience perhaps that want to see a dietitian who have IBD, where should you direct them to, or how can I access someone?
Yeah, no, that's a really good question. So there are a couple of organizations that you can rely on because they are, so the Deacan organization, DECCAN, and I can put that in the chat as well for some extra information. That's where pretty much all IBD dietitians or IBDians that also work in IBD register. And we also have monthly meetings where we keep each other's up to date, and you can find them across all Australia pretty much. The other place to go to is Dietitians Australia. So there you will be able to check where to find a dietitian, and you can check the speciality of each dietitian. And there is a box that you can tick specifically for inflammatory bowel disease. So at least they can point you to someone who has expertise in the area.
Thank you. Thank you very much.
You're welcome.
Thanks, Chiara. We might move on almost to our next speaker, but before we do that, I'd like to set this up for you, Denelle, by answering one of the questions that has been posted. So, George, can I ask you to kind of answer the question about, do we know what a healthy gut looks like? And, you know, what can we do to shift it to a healthier profile?
Yes, absolutely. It's if I knew the answer, I'd be very rich and I wouldn't be here. Okay, so you know, at the end of the day, we're still figuring out what healthy looks like. And if I give people an example, we use fecal microbiota transplantation to improve the health of the gut microbiome. It works very, very well in some conditions, such as Clostridioides difficile. It doesn't work so well in inflammatory bowel disease. But if I was to, if we accept the concept that some people are healthier than others, and we know that because some people have disease or not, if we were going to use a fecal microbiota transplantation for improvement of health, and I gave you two options. I give you a 19-year-old healthy chap who, you know, student, and I give you, you know, so superb physique, athlete-wise, and I give you that as a choice, or I give you a 62-year-old chap who probably drinks a bit too much, but has a little bit of a paunch. Which one of those are you going to choose? I'm assuming
probably the healthier one.
Absolutely, the younger one, right?
Yeah.
But that younger one hasn't seen anything of life. The older gentleman's got to 62 years old without developing any significant disease, and does drink too much, but actually his body's still okay with that. You know, so he's kind of tried and tested, but it's not what you would automatically choose. So, you know, it depends on what your concept of healthy is. It depends on what you decide to measure it on. But what we know so far is that diversity is good. We know that, you know, if you can do anything, it would be about understanding what you eat and how that influences your microbiome, and, you know, diversity in what you eat. You can't just eat the same thing again and again and again because it's healthy. It's about asking different questions of your microbes. So if you change the nutrients, your microbes will respond differently. If I had a tuna sandwich every day, I'm going to get pretty bored of it, and I'm going to turn off my sensory, you know, my nose is not going to enjoy the smell of it anymore. So give me something different. Wake up my senses, wake up your microbes is what I would say. Eat the rainbow. That one.
Yeah. And for the audience, there is evidence that eating 30 different plant foods per week results in a gut microbiome profile that's, we think, potentially better, and it's associated with healthier people. So there is lots of merit to that. Okay, so just in relation to that, I will move on to you in a second, Denelle. Could you, George, maybe talk about the home study then, and just what the rationale is for that study that you're doing?
Absolutely. So it's another study that's running out of the Microbiome Research Centre at University of New South Wales. I'm not directly involved in it, but I can triage any questions that you might have. But essentially, it was to really define what the microbiome of the healthy optimum Australian microbiome. So to look at what is the healthy optimum microbiome in Australians. And so the concept really was, people of different sort of ages, and particularly older sort of active centenarians, there must be something about their microbiome that is helping to keep them healthy. So comparing against people, for example, who are ultra fit, ultra athletes versus children that are doing lots of activities as well. So they're actually sampling many different groups of individuals, as well as people who have developed disease such as neurological disorders, to actually be able to understand what it is about microbes that actually is keeping people healthy, so we can understand the ones that we need to hold on to. And maybe supplement. And so the home study, and I apologize, I don't have the QR code with me. I should have done, I'm so sorry, but you can scan through on my QR code and I can pass that information on. But they're really looking for people that fall into those categories of, ultra healthy, ultra old, various different categories, to understand what it is about in Australia that we need to hold dear and true to remain healthy.
It's fascinating, isn't it? So that's currently still recruiting, and if people are interested.
Still recruiting, yeah. Absolutely.
Okay. All right, well, thank you. So we might hand over now to Denelle, if you'd like to share your presentation. So Denelle's research explores the relationship between the diet, gut microbiota, and inflammation, specifically in adults with IBD.
Thanks, Kelly. Very thrilled to be sharing with you guys a little bit about my PhD research. So when we think about diet in the context of IBD, as Chiara mentioned, it's something that people living with IBD are incredibly interested in. And what the evidence tells us is that different diets are associated with how likely someone might be to go on to develop IBD. And we have two pictures here, and one can be used to represent a Western diet. And lots of research from different populations around the world, and quite large studies, have shown us that this following or eating this type of Western diet places people at a higher risk of developing IBD. Whereas oppose that to the healthy diet shown on the right, which is typically shown to result in a lower risk of developing IBD. So diet is implicated in developing IBD. But when we think about the role of diet in managing IBD, once someone's already developed it, there's a lot less information known. So lots of different therapeutic diets have been studied for whether or not they can promote remission and lower inflammation. For example, those diets on the screen, so specific carbohydrate, paleo, vegetarian, Mediterranean are just a few. But what we know is that, so lots of these diets have been studied, but there is no conclusive evidence that any one particular diet is best for people with IBD to follow. And so that means that there's not one of these particular diets that is like guaranteed or definitely going to help someone reduce inflammation. And so where we're at right now with the research is that it's the general Australian dietary guidelines and general healthy eating patterns, which people with IBD are recommended to follow, like the rest of Australians. But we know that emerging evidence has started to show that there is this relationship between what we eat, our microbes, and inflammation. So most of the foods we eat end up in our colon, our large intestine, and depending on what we eat, depends on which bacteria ferment and feed off that food. They feed off it and they produce gases and metabolites. Now, certain bacteria produce these metabolites which are beneficial for our inflammatory or our immune system. And then other microbes ferment different types of foods and produce these metabolites, which can actually kind of increase the levels of inflammation in our gut and just not be helpful in managing a healthy immune system. So obviously, when it comes to a disease like IBD, it's of great research interest to try and understand a little bit more about how these three diet, microbiome, and inflammation are related. And because of this, and as Georgina mentioned, lots of probiotics, for example, have been quite well studied in IBD as a microbiome therapy or of a bit of a nutritional therapy. And probiotics can induce remission, so they can bring people from higher states of inflammation down, particularly in ulcerative colitis. Prebiotics, which are defined as so the foods that we eat that are fermented by our microbes and feed them, So these prebiotics might reduce inflammation and they might change the microbiome. But there's a couple of gaps in our knowledge about this kind of area of research. And the first is that this reduction in inflammatory markers is not clinically significant. So when you have someone who has Crohn's or ulcerative colitis, and you take a blood test, they often have really, really high inflammatory markers. And these dietary therapies have kind of shown to bring that down by maybe 3 to 4 points. And that's just not significant when that level might be upwards of 200. And additionally, in a lot of this research, the microbiome and the inflammation outcomes have been studied separately. Particularly of an area of research interest when we think about microbiome therapies in inflammatory conditions is this category of plant bioactives called polyphenols. So polyphenols, a lot of them are responsible for the different color components in fruits and vegetables. And a particular type of polyphenol that we're super interested in is this anthocyanin. And that's responsible for that deep purple blue color seen represented in that image on the side. And anthocyanins have shown the potential to feed gut bacteria, so act as those prebiotics, and to reduce inflammation, but in other human populations. So they haven't really been studied for whether they have the same effect in adults with IBD. So that brings me to kind of summarizing a little bit about there's some missing information from this area of diet microbiome inflammation relationships. And that is that we don't have a lot of research showing us what dietary therapies can do to both the microbiome and inflammation. And if we want to kind of understand how our microbiome influences inflammation, we need to test these two at the same time. We need to be able to draw some relationships between those two outcomes. As I mentioned, some therapies have shown the ability to reduce inflammation, but not powerful enough. We need more powerful anti-inflammatories to work alongside medication and support people who have high levels of inflammation. And additionally, we really want to know what is the role of polyphenols as prebiotics and anti-inflammatories in IBD. So to address some of these missing bits of information, one of my PhD studies is called the INHABIT Inflammatory Bowel Disease Trial. And the INHABIT stands for Synergistic Effect of Anthocyanins and Probiotics. So what we're doing in this study is we're supplementing people with freeze-dried blackcurrant powder, which contains really high amounts of that anthocyanin compound, and a multi-strain probiotic. And we're testing a lot of data. We want to know whether or not these nutritional supplements can improve their quality of life and reduce some of the bowel symptoms that people with IBD fairly frequently experience. We also want to know whether or not these therapies can reduce inflammation in the stool and in the blood. And we're collecting detailed data on the gut microbiome to see whether these therapies can change the microbiome and how it functions. So we're expecting that this clinical trial will help to provide some missing information on how anthocyanins can influence inflammation of the microbiome on their own and then with a probiotic. And we want to get some more information about how inflammation and the microbiome are related and work together. And for example, do we see patterns in the changes in the microbiome in people who do see a big drop in inflammation versus those who don't? And all of this kind of ties into what Georgina was talking about earlier about precision medicine and precision nutritional therapy. We need more information about how the microbiome works if we're going to be able to provide targeted and individualized nutritional therapies. So who we're looking for in this study is 100 adults with ulcerative colitis and on the mild to moderate end of the spectrum, and people aged 18 years and over. And what our participants will be doing, they'll be taking a blackcurrant powder and a probiotic for 3 months, or the placebo form of those. They'll be providing stool and blood samples at 2 visits. And before and at the end, and then in between, also completing some online surveys, looking at health, what they've been eating over the past couple of days, and some cognitive and mood assessments as well. And so we can see a bit of an overview here of our study timeline. So we've got 4 different arms to this trial. People are randomly assigned to one of these arms, come in at baseline, they give stool, blood samples, we ask them about diet, about their gut symptoms, about cognition. We repeat some of these measures at 6 weeks, and then we repeat all of the measures as well as the stool and blood at 12 weeks. And that's to see how much our products have influenced these outcomes. And the really exciting thing is that we are recruiting for this trial. So if you have ulcerative colitis, if you have a family member, a friend, a work colleague, please let them know about this study. You can, if it's you listening and you have ulcerative colitis, if you scan the QR code, it'll take you to a screening survey to test your eligibility. Or if you want more information to, say, pass on to a friend or something, you can get in touch with me via that email and that phone number, or just if you'd like to know more about our study, we'd love to hear from you and have you involved. Thanks for listening.
Thanks, Denelle. Thank you very much. I have two questions for you, if I can.
Mm-hmm.
We have one question that has asked, how do you actually measure stool inflammation?
That's a great question. And I believe Georgina might have mentioned it earlier, but the particular outcome we're looking for is called fecal calprotectin. And it is a similar to when you might go and get a blood test and we send it off and someone processes it and they tell us these levels of inflammation in the blood. We get people, our participants, to give us a stool sample. We're sending it off to a biochemistry lab who then analyze the stool, and they find this intestinal marker, and it tells us how much inflammation is in specifically in the intestines. So when you look at blood inflammation, it's telling us your systemic inflammation, so how much is kind of in your blood. But this fecal calprotectin is really, really accurate at detecting inflammation directly in someone's colon, large intestine.
Lovely. And my second question is about the concept of inflammatory foods. Are there really inflammatory foods, or is it more about dietary patterns that makes things inflammatory?
Great question, Kelly. And I love the way you phrase that. Yes, it's much more about, think about food, habits of food or dietary patterns rather than one single food itself. So research has shown that there are certain ways of eating that increase someone's likelihood of developing inflammatory conditions like IBD or increase their inflammatory markers. And that's more what we would consider, what I kind of describes as that Western pattern has more processed foods, foods more likely to have saturated trans fats, added sugars, lower in the nutritional content. And so eating those foods often and in combination with each other, a majority of your diet is much more likely to put someone in a position of increasing susceptibility to these inflammatory conditions. But it's important to know that, if you eat a healthy, balanced diet, reasonably following, Australian dietary guidelines, and you have like a burger every now and then, when you eat that, that doesn't immediately increase inflammation. It's more about how often and how much. Yeah.
Excellent. And I think I might answer the other question that's been put in the chat about the role of a low FODMAP diet. So look, inflammatory bowel disease is different from irritable bowel syndrome, but there are a good chunk of people, around the 1/4 to 1/3 of people who have inflammatory bowel disease who also have coexisting in irritable bowel syndrome. And for that cohort of people who don't get remission from their symptoms, perhaps there's a role for a low FODMAP diet. So this is a specialized type of diet where you may be susceptible to certain chemicals naturally found in food, and they cause fermentation in the gut and gut symptoms, and it can be very problematic for people. But not everyone with inflammatory bowel disease would benefit from that. And certainly you don't need to eliminate all foods rich in FODMAPs. It may be just specific subclasses of FODMAPs. So if you do think that you might fall into that kind of category of somebody who's got inflammatory bowel disease and you just got a grumbling gut all the time, you're not seeming to get some symptom relief, then perhaps you might be the sort of person that might benefit from a trial of a low FODMAP diet, but do it under the supervision of a dietitian, because they're not easy diets to follow and they're not nutritionally complete either. So you do need some supervision. Now, I'd like to kind of start wrapping up by asking people to give us perhaps some key messages that they would like people to take away when it comes to gut health. Perhaps, George, could I start with you? Are there any take-home messages that you'd like people to kind of walk away with knowing?
I think as an active researcher within gut health, the thing that I would say is we know an awful lot, but there's an awful lot more that we need to know, and we really need people who have an interest to actually step up and be part of the studies that are going on. If they are, it takes a long time to do these types of studies, and the more people that are part of the research, the more of a comprehensive understanding that we will get. So that's the first thing I would say is own the research so that we can move things forward. Secondly, as we talked about before, health is a very personal thing. And there is so much that we can do if we just try to take some action to be as healthy as we can be. That's difficult, and it's not the same for everybody in any shape or form, but the more we can do to keep ourselves healthy, by whether it be a healthy mind or a healthy diet or a healthy body, the more it's going to pay dividends in the long run. We have a very overstretched health service, and we are an aging population. And all of these factors mean that in the future, the way that we access healthcare is going to need to change, because we can't sustain what we have. And so the more that we can do to take responsibility for ourselves, the more we're going to be the health service, and we're going to be around for longer.
Yeah, thank you. Chiara, Denelle, any key messages you'd like people to take away?
Hey, thank you. I'll take 2 seconds. Firstly, I second what Georgina said. It's important that you keep your diet as varied as possible, and that you do participate in studies, because we always struggle to find participants sometimes, and we understand people's time is precious, but it's always very much appreciated, because you're not just helping others, but ultimately you're going to help yourself as well, because we are further our knowledge and your knowledge too. The second thing I like to say is that in terms of gut health, there's so much stuff on the internet, and that people can be talking about. It can make things really confusing. So if you do have questions, don't be afraid to reach out to maybe your GP or your gastroenterologist and advocate for yourself and say, I would like to see a dietitian that, works in IBD, because I have some questions I would like to address. And, we all aim for person-centered care, which is, from a clinician point of view, we want to do the best we can to support our clients. And so whether the gastro or the GP might not agree with you, because they have very different views about the role of diet in IBD, they still need to listen to you and provide some advice. So don't be fearful of asking those questions. Just go ahead and ask.
Thanks, Chiara. I would say that I kind of touched on this, but what we eat does have a big role to play in our gut health. And there's a lot of although it's a new area of research, there is lots of research always coming out to support this. And we know that the microbiome is really quite responsive to dietary changes. And so something I would recommend is, as Kelly mentioned before, increasing the amount and the diversity of plant foods. So fruits, vegetables, whole grains, nuts and seeds, legumes, lentils, herbs and spices. The more of those different foods that you have, that's feeding and helping to kind of culture a really healthy, flourishing microbiome. And in a lot of instances, generally in the healthy population, those dietary changes have shown more benefits in a lot of cases than supplements. And so you might not need supplements. And, there's, Chiara mentioned a lot of advertisement about what's gut healthy supplements and powders and probiotics. Focus on your plant-based foods, focus on having fermented foods, particularly fermented dairy products daily, and trying to eat a dietary pattern that's more closely aligned with the dietary guidelines or a Mediterranean style eating pattern.
And so then, is there still a role, or should there be a role for adding a probiotic supplement on top of that if you're already healthy?
Yeah, I would say so, definitely. From thinking about a dietitian perspective in gastrointestinal diseases, I think seconding what someone said earlier, often we will look at specific strains to, for example, manage symptoms if someone has like an irritable bowel syndrome, inflammatory bowel disease as well. But generally, I would say a diet first approach, and then we look at, well, what can we supplement with that? Is there a role for like a fiber supplement or a probiotic supplement as well to boost those health benefits?
Yeah, it definitely requires advice and guidance, doesn't it? And taking one off the shelf is probably not going to help everybody. It's used in a targeted way. Any other questions from the audience? Please feel free to put them in the chat now. And if not, we shall answer any questions after the presentation today. Feel free to email us. So without any further questions, I think we might wrap this up here. Thank you to the audience for engaging with the questions and answers, and hopefully you've learned something else today. The event has actually been recorded, so a recording will be sent to you after this. Thank you very much, and hopefully you can join us for some more of the Luminary sessions in the future. Otherwise, thank you again, everyone, and have a good evening. Thank you.